Hello microbiology students, and welcome to chapter 1 of our textbook, Microbiology Fundamentals. This chapter is going to focus on an introduction to microbes. If we look at the chapter outline, you'll see it has 4 sections that are in our text. However, we are only going to cover sections 1 and 4. I am removing sections 2 and 3 because we are not required to cover that based on our Technical College System of Georgia State standards. I teach by our standards, not necessarily to the text, because the text very well may and does have extra information in it compared to what our state standards are. So please keep that in mind throughout the course of this chapter, as well as future chapters, that you should utilize these PowerPoints to help you to narrow down your content for the purpose of studying for exams. My exams questions come from the PowerPoints, not the text. So if it's in the PowerPoint, it's testable. If it's not, then it's not. Alright, so starting with section 1, microbes. The field of microbiology is by definition the study of things that are microscopic. And so what that means is that microscopic things are too small to be seen with the naked eye. So we have to utilize some kind of tool that increases the resolving power in order to see these objects or organisms. And we use a microscope to do that, which is a chapter or a section for another video. So again, microbiology, if we break that down, micro meaning small, bio meaning living, and the ology meaning the study of something. So altogether, microbiology is the study of tiny living things. Some of these living things are cellular, and so what that means is they are made up of cells. This includes bacteria, fungi, protists, and they're an example of a few animals that are microscopic that can infect humans, and those are the helminths. Helminths are parasitic worms. They technically can also be macroscopic, meaning you can see them with the naked eye. However, the way we detect parasitic infections of worms is typically through finding the eggs or larvae in a sample, and those are microscopic. So for that reason, they are still a part of microbiology. So again, the following organisms of these categories are cellular: bacteria, fungi, protists, and helminths or parasitic worms are cellular organisms that can infect a host. Oppositely, some of these items, or some of the items in this, or these items below in the list, are considered acellular. A, when it's at the beginning of a word, typically is going to mean the opposite of whatever the root word is, in this case, cell. So these two guys down here are acellular. They are not, this is my symbol for not, made up of cells. And those include viruses and prions. So this picture here comes from our text. It lists out the various microorganisms that we have discussed in this list here and describes them as either acellular or cellular. So as we can see, viruses, prions in my list here are acellular. And the cellular organisms include things like bacteria and eukaryotes. The following, fungi through animals, are eukaryotes. I'm going to write E for short because I'm out of space. But we will talk again about that in the future, possibly in this PowerPoint. If not, then in a future one, about what the difference is in eukaryotes versus other organisms. And some of you may already be familiar. If you took Bio 1 with me, then you should know what a eukaryotic organism is. But we'll come back to that later. Alright, so in fact, actually, we're going to talk about it now. Please forgive me. This is my first time making a video on this using this PowerPoint, so the order of operations here I still haven't memorized. But Organisms such as microbes, especially things like bacteria, are single-celled organisms, and they appeared on the planet about 3.8 billion years ago. And if you look at this tree of life here, what this shows is the phylogenetic history of evolutionary life. This tree, which kind of looks like a tree where this would be the trunk. Oop, my line's not straight here. The trunk, and we see the branches coming off, and more branches and more branches and so on. This very initial point here, this trunk of the tree, represents the last common ancestor, meaning the very first living organism on Earth. And again, that arose about 3.8 billion years ago. And the various cell types that exist today arose from that single extinct common ancestor. So now we have three categories. We call these domains of life. That includes bacteria, archaea, and eukarya. Eukarya, this domain, includes groups of organisms whose cells are eukaryotic. And what a eukaryotic cell has that sets it apart from other cell types is it has a nucleus. And when we say a nucleus, y'all should be familiar with that from a prerequisite course, either A&P or Bio 1. But it is basically the command center of the cell, and it houses the DNA. Other groups of organisms found in domain bacteria and archaea are considered prokaryotes. Pro meaning before, and karyo meaning nucleus, so before the nucleus. There is no nucleus in prokaryotic cells. So these two groups, these domains, include cells that are prokaryotic. And this domain will include organisms that are eukaryotic, that have a true nucleus, versus the other group, which is prokaryotes that don't. The following picture at the bottom also gives you a little timeline of events here. Again, we have our last common ancestor, which was arose about 3.8 billion years ago. You can see the probable origin of Earth was about 4.6 billion years ago. But from that last common ancestor, we had bacteria exist about 2.8 billion years ago, as well as archaea. Those kind of came very close together in time. More recently, we see about 6 billion years ago, we see our first eukaryote cells. Multicellularity happened much later on, more recently. In fact, insects we see here was less than 1 billion years ago, as well as reptiles, mammals, and humans. So, what this is demonstrating is that microbes are very old. We've seen these exist on the planet, or I guess I can't say really seen them exist, because that would obviously we didn't exist that long ago, but we have evidence to support that these single-celled organisms existed 3.8 billion years ago. And these are the primary organisms: bacteria, sometimes archaea, and various eukaryotic cells like fungi, or helminths and protists that can infect other organisms. Our next topic is microbes are ubiquitous, and ubiquitous simply means everywhere. They are found in the deepest areas of the Earth's crust. They are found in polar ice caps. They're found in oceans. They're found inside the bodies of plants. They're found in the bodies of animals. They're all over the place. They're in the Earth's landscape, and basically they're essential to life. I know we like to associate bacteria with disease, especially considering this is a clinical approach to the course, considering most of y'all are wanting to go into some kind of clinical profession. However, in the grander scope of things, bacteria are essential to life. They help control the ecosystem of Earth. We would not be able to digest certain things if we didn't have a normal bacterial flora in our GI tract. And just to demonstrate how ubiquitous bacteria really are, I have this picture over here to the right. So what was done here is we took an agar plate. I'll write that down, AGAR. An agar is this somewhat of a jello consistency. Think of it like a hard jello, sort of. It's a little more solid than like just the jello you would eat, but it's still a you could definitely squish it if you wanted to. But this agar has oftentimes nutrients within it that has been put into it by some kind of manufacturer to promote microbial growth. So we allowed someone's hand to sit on this agar, and then we removed it. Then I put a lid on this agar plate, and I let it sit for a period of time, we'll say approximately 2 days. And this is what we saw after that. We see tons of microbial growth. Each one of these little circular shapes, this one, there's so many you can't even count all of them, but all of those are colonies of microorganisms, most of which are bacteria, but I do suspect some yeast or molds on here as well. But that just goes to show you how ubiquitous these microbes are. So next, how do we use microbes as humans? So there are several different examples of historical uses of microbes in various ways, and most of these we still use today, like in bread production, and alcohol production, cheese production. Oftentimes these are going to be done through either yeast or bacteria. Cleaning up human-created contamination even. There's been bacteria that has been genetically modified to break up oil spills and clean them. That's pretty cool. Now, what about the human harm? So, like we said, not all bacteria are harmful for humans, and in fact, they are really important to life. The vast majority of these microorganisms that associate with humans are harmless, and many of them are even beneficial. Bacteria are a good thing. But about 5% of the bacterial species are what we call pathogenic, and you need to know what that means. Pathogens are any microbe that can cause a disease. There's over 2,000 different microbes that can cause disease, and 10 billion infections occur across the world every year. And these infectious diseases are an important common causes of death worldwide, hence why we're interested in them. So if we take a look at this graphic from our text, I thought it was pretty interesting to look at because it shows you the top causes of death compared to the United States versus the world. So what we're seeing here is globally, the heart disease is the most prevalent cause of death both worldwide and in the United States. Afterwards, we see cancer. We see stroke as the second leader of death worldwide. COPD, accidents, lower respiratory diseases, and again, lower respiratory diseases here. I want you to take a look, though, at the ones in pink. And notice that the ones in pink are caused by an infectious disease. See, if we look here, this pink represents diseases that are clearly caused by microorganisms. The ones in blue are not currently known to have an infectious cause, meaning it caused by a microorganism that can be spread from one person to the next. But I think this is interesting to see here because we see that the top causes of death in the United States for infectious diseases is relatively low. The most common cause of infectious disease death in the United States is influenza and pneumonia, compared to the world, which is going to include lower respiratory tract infections, diarrhea, and tuberculosis. Alright, so what are microbes exactly? So like we mentioned previously, some are cellular and some are acellular. We'll start with the cellular ones first. Our eukaryotic cells, again, have a nucleus. If we look at this picture here, it's going to show you various organelles, and that should be familiar to you from A&P or from biology. And these eukaryotic cells have various organelles. Think of organelles like tiny organs inside of a cell. They are typically membranous structures that has a structure similar to the plasma membrane, and they're going to perform specific functions. So, for example, the mitochondria is a major site of ATP production. This is where we get energy. Chloroplasts. These are found in plants, some protists, plants and some protists, and algae, which is technically a protist. But anyway, these chloroplasts are capable of converting solar energy into chemical energy through a process called photosynthesis. Some other examples of organelles are shown throughout. We see the Golgi complex, lysosomes. We see the rough endoplasmic reticulum, smooth endoplasmic reticulum. So hopefully those ring a bell from our prerequisite classes. But anyway, some organisms that are considered eukaryotic are microorganisms, namely, for the most part, protists. And some are macroscopic, like you and me. We're eukaryotic organisms that are multicellular, and we can see each other. Prokaryotes, on the other hand, include the bacteria and archaea. They do not have a nucleus. So if we compare this illustration of a eukaryotic cell to this one, which is a prokaryotic cell, could be bacteria or archaea, we do not see the nucleus in this cell because there isn't one. The nucleus in the eukaryotic cell is here. It's a membrane-bound organelle. Almost think of this like a bubble inside of the bigger bubble, which is the cell membrane. Instead, the DNA is found in the nucleoid region. So we can see all of this blue spaghetti looking stuff. That is the DNA, and it's just going to be found dispersed throughout the cell in a region called the nucleoid. It is not membrane bound like it is inside of a eukaryotic cell. Bacteria and archaea, our prokaryotes, are typically about 10 times smaller than eukaryotic cells. We don't see all these specialized organelles inside of our prokaryotic cell, which there's some exceptions, like they do have ribosomes, just like our eukaryotic cell does here, but most of them are absent. So despite how simple these prokaryotic organisms are, they're an extremely successful group of organisms. It's kind of interesting to know, in my opinion, that if you were to count all of the bacterial cells in your gut flora, there would be more bacteria, or count all of it in your gut flora and the ones that are on you. So basically all the bacteria in and on your body, there would be more bacteria cells than there are you cells. So it's kind of interesting to know. And then it gives you just another rendition of how ubiquitous or everywhere these organisms are. All right, so we said some of these microbes are acellular, and again, that means they do not have a cell structure. That includes our viruses and our prions. Viruses are not independently living cellular organisms. Oftentimes you'll hear them called intracellular. I'm going to write it down. Intracellular parasites. May even hear the term obligatory. Obligatory intracellular parasites, which means that they have to invade living cells in order to carry out some of their processes. And essentially, they hijack cells and their cellular machinery, like the ribosomes and the other organelles, to make more viruses as part of their reproductive process. So they exist at the level of complexity somewhere between large molecules and cells, but they are not considered cellular organisms due to the fact that they lack a cell structure and cannot carry out their life cycles independently without using a host cell. Viruses as a whole are composed of a small amount of hereditary material. This can be DNA or RNA, and it's surrounded by a protein coat and sometimes a membrane. And we'll get into more details on that in another chapter. Prions are even simpler than viruses. They don't have any DNA. It's only a protein. It's an infectious protein. They act like microorganisms even though they're not considered an organism. They're not a living thing, and they don't even have any genetic material. So here's 6 types of microorganisms that are shown under a microscope. Here we see a helmet. This is an example of a parasitic worm. This would be the head of it and the body. Can't see the whole thing because it's too far blown up. This is a fungus. Specifically, this one's called mucor. And we'll talk about all these structures in another chapter. Here we have a protist called Vorticella. Each one of these little bell shapes is a different organism, a different individual, Vorticella. We see in this picture bacteria, all these little tiny, they almost look circular, but they're really rod shaped. We'll get into that later, but those are bacteria. Here, each one of these little round structures are viruses. And here we have prions, which are, again, an infectious protein. All right, so we're going to go into more details on those different types of classifications of microbes in the next few chapters. But before we do that, we're going to finish out Chapter 1. And Chapter 1's last section is about naming, classifying, and identifying microorganisms. Based on my experience with teaching this class, I feel like this is probably going to be the harder part, especially if you took A&P rather than biology. So please don't hesitate to reach out to me if you have any questions or need any kind of clarification on anything. But because there's so much diversity amongst living things, there's an entire field of biology that is dedicated to the science of identification, classification, and naming of organisms, and that's called taxonomy. Taxonomy. The goal of a taxonomist is to first and foremost identify an organism. Identification is the process of discovering and recording the traits of an organism so they may be recognized or named and then classified. So this is a kind of a long definition of basically saying if I see a lion, or let's say a zebra, how do I know it's a zebra? Well, they have certain characteristics that we can utilize to identify them as a zebra. So a taxonomist's goal is to identify living things based on recorded traits like white and black stripes, etc., so that we can, if we see another organism that looks just like it, we can safely provide evidence that it is most likely also this zebra or whatever other organism we're referring to. The next goal of a taxonomist is to classify. Classification is the orderly arrangement of organisms into a hierarchy. So, for example, what defines an animal as an animal? What defines a plant as a plant? What defines a protist as a protist? What defines a chordate? These are all examples of classifying organisms, but in taxonomy we classify them in a hierarchy based on evolutionary relatedness, and I'll expand on that momentarily. But the last job of a taxonomist is nomenclature. Nomen, that root word there, is literally, the Latin translation is name. It's name in Latin. Nomen, name. So it's to give organisms names. It's the assignment of a scientific name to various taxonomic categories and to individual organisms. So let's expand on what we mean by this hierarchy business we've been mentioning. So currently we utilize an 8 nested hierarchical categories to classify organisms. So there's 8 hierarchical categories. We call these taxa or taxon. Taxon is singular, taxa is plural, that are nested within one another. They are as follows: domain, kingdom, phylum, class, order, family, genus, species. You need to know those in order. My mnemonic device to try to help you remember those are listed here. All right, so Dear King Philip cried over family going to Switzerland, where each one of these words will coincide with the hierarchical taxa. So dear, domain. The K in kingdom, the K in king stands for kingdom. The P in Philip stands for phylum, and so on. At the very top, this top hierarchy, this top taxon, domain, is the most inclusive. And we get more and more specific as we get down. And as we get to the very last one, the species level, is the least inclusive because it includes only one species. So let's look at an example. So domain. All of the organisms listed here are in domain Eukarya, which again means that they are eukaryotes. They have a true nucleus in their cells. If I want to then order or list out the organisms that are in kingdom Animalia, then all of these same organisms would be listed except ones that are not animals. So we can see dog is in Animalia. It's also in Eukarya. Wolf, coyote, fox, lion, seal, mouse, human, whale, bat, fish, snake, earthworm, and moth. But paramecium and tree, they are eukaryotes, but they are not animals. Paramecium are a protist, and a tree is a plant. So we do not see them listed. We're getting more specific as we go down. So if we look down to the next level of organization, which is phylum, this is showing all organisms that are in phylum Chordata, which means, to make a long story short, they have a vertebrae. They have backbone. So we'll see all of the ones that are listed here are also found in Animalia, except for the ones that don't have a backbone. So dog, wolf, coyote, fox, lion, seal, mouse, human, whale, bat, fish, snake, but earthworm and moth, they do not have a vertebrae, so they are not considered chordates. They are not in phylum Chordata. Now, in Mammalia, we're only listing those that are mammals. Fish and snake, not mammals. Then we have order Carnivora. We see that whale, bat, mouse, and human are not listed in Carnivora. They are not carnivores. Yes, humans eat meat, but we also eat plants. And then we have family Canidae, genus Canis, and species Lupus. There we go. Which includes only dog and wolf. Alright, so this is a picture from your book just to give you another example. So again, our classification is organized into taxa or taxon. Taxa is plural. And those are domain, kingdom, phylum, class, order, family, genus, and species, with domain being the most inclusive, including the most organisms, and then species being the most specific. So the examples they give you in the book is shown here in this image. We see domain Eukarya. So this is all eukaryotic organisms would be in that domain. As we move down to the next level, we see kingdoms. So in kingdom Animalia, we see things like, I believe that's a sponge possibly, or a coral. We see human. We see, I think that's a lemur, starfish, dog, and gorilla. However, over here in kingdom Protista, we see various examples of protists. Difficult to say what they are just by looking at them. I can say that's a paramecium. But the other ones I can't be as sure. As far as phylums, Chordata, this includes all of the organisms that have that are going to have a vertebra. So you'll see less and less organisms that are going to fit into those categories as we get down to the species level, which will only have one. Likewise, over here we see the phylum Cleophora, and those are only protozoans that are going to have cilia. And we're going to get more and more specific as we go down until we're at the species level and only are pertaining to one type of organism. Now, this ties in with nomenclature because the taxonomic system we use utilizes the genus and the species names to produce the scientific name. So scientific names are written out as genus, then species. We always want to make sure that it's italicized or underline it if you are writing it by hand. Italicize if you're using a computer. And when the name is abbreviated, the genus name is abbreviated to the first initial followed by a period and the full species name written afterwards. So, for example, here we see handwritten Escherichia coli. You probably are familiar with this. If I abbreviate it, it would be E with a period and then coli. Notice that the first letter is capitalized in the genus, but the species is not. Alright, so next I have a video from the Amoeba Sisters. You'll see I like to post these. If they're not my PowerPoint lectures, they're in my, I'll post them to your Blackboard. But this is about naming, classifying, and identifying, microorganisms. So I'm going to play this last.
There are a lot of animals that I have always thought are cool. Salmon, hairless guinea pigs, iguanas, silky bantam chickens. But one day in my 7th grade science class, my teacher told us that we would get to see one of the most amazing animals ever in class, a hydra. And I have to confess, what I imagined in my head and what was actually reality was a little different, but I'm kind of used to that. Turns out hydra are pretty cool. Hydra are animals that are very small, a few millimeters in length, and they live in fresh water. They can viciously attack and eat their tiny swimming prey, and they can reproduce by budding an identical offspring off of themselves. But if you saw them, you might not think they're an animal. What makes us categorize them as an animal anyway, and not a plant or a protist or a fungus? In fact, how do we classify in the first place? Well, this lets us jump right into our topic, classification. Taxonomy includes the naming and classification of species. And much of the credit for starting a formal classification goes to Carl Linnaeus. We'll get back to that a little bit later in this video. But back in his time, the 18th century, there wasn't a separate category for other organisms like protists or bacteria. They didn't know about detailed cell structure differences. They didn't know about DNA, so they didn't know genetic relationships that now can determine how we categorize organisms. And that's the thing about classification. As we learn more about DNA, and therefore relatedness, how we classify organisms can continue to change. So first, let's look at an awesome hierarchy system mnemonic. There's a lot of great ones that you can find that people have shared online. We'd also love to share ours. Dear King Paramecium cares only for green spirulina, but we sort of have a thing for protists, and we realize that might not be as memorable. So let's take a look at each group of this hierarchy, starting with one of the most inclusive groups, domains. Domains. It's so awesome that all of life will fit into them. So there are 3 domains, bacteria, archaea, and eukarya. The domain bacteria is full of bacteria. They're prokaryotes, and therefore they have characteristics we've mentioned of prokaryotes before. These can include bacteria that make you sick, the bacteria that are in your intestines helping you digest, the bacteria helping with decomposing, the bacteria fixing nitrogen in the soil, tons of different kinds of bacteria. Archaea are prokaryotes too, but they have some major DNA and structure differences that give them their own domain. And while they may seem more closely related to bacteria since they are prokaryotes, recent DNA evidence links them to having more in common with eukaryotes, which is interesting. Many of the archaea are extremophiles. That is, many of them like the extremes. Some like extreme salt environments, for example, which means they can handle extremely salty environments like the Dead Sea. Or methanogens. They can live where there is very little oxygen. In fact, most of them can't even handle oxygen. They use carbon dioxide to make their energy instead and produce waste gas, methane. Some of them live inside animals that eat a lot of cellulose, like cows or termites. Another extremophile is thermophiles. They like extreme temperatures. If you ever dreamed of living near the deep-sea hydrothermal vents, well, if you were a thermophile, you'd be in luck. We mentioned that archaea and bacteria domains are separate because they have some major DNA and structure differences that are significant enough to separate them. And so does the third domain, eukarya. These are eukaryotes, and so they have characteristics that we've mentioned before that are common for eukaryotes. And that's where we'll focus right now. So the next level, less inclusive and more specific than domains, is the level of kingdom. Now here is the big disclaimer about kingdoms. Its organization is often changing, and it's not even something that all scientists agree on. We've seen a 5-kingdom system that looks like this, and a 6-kingdom system that looks like this, and it's important to understand it's a changing view as we learn more about DNA and cell structure evidence. That's what makes classification so exciting. But if we focus on these eukaryotes, let's touch on them briefly. Protista, extremely diverse, and there's often talk about dividing it because of how diverse it is. There are protists that are animal-like, and protists that are plant-like, and protists that are fungi-like, but many scientists don't consider them to quite meet the requirements to be in those kingdoms. Protista includes both autotroph protists, making their own food, and heterotroph protists, which can consume other things for energy. Most protists are unicellular, but they can be multicellular. Some have cell walls made of cellulose, which is kind of similar to plants, and some don't. Fungi are heterotrophs, so no photosynthesis for them. If that's hard to remember, just think about athlete's foot. It's a fungus on your foot, and it's not doing any photosynthesis there because that'd be really weird. Nope, it's there, causing irritation, eating dead skin cells. Fungi are usually multicellular, but they can be unicellular, and most of them have cell walls of chitin. It's a carbohydrate. Next up, plantae. Plants are autotrophs. Yes, even the carnivorous plants, because they still make their glucose from sunlight energy. Plants are, for the most part, multicellular, and most have cell walls of cellulose. Finally, last up, animalia, animals. Okay, Hydra, you can come back. This mostly multicellular and heterotroph kingdom is the kingdom to which you belong. So now we have the other hierarchy levels. We get less inclusive, therefore more specific, as we move down to the Hydra's phylum, class, order, family, genus, and species. Because the species name is the most specific that you can get, it is the least inclusive. Now, you'll notice we wrote its species name here. Remember how we said we'd bring up Carl Linnaeus again? Well, it's because of him that we have this naming system, binomial nomenclature. This 2-part naming system that we use uses Latin or Greek roots. This is its scientific name. See that first name? That's its genus. It's written with a capital letter at the start, and it's written in italics. See the second name? That's its specific epithet, which is a fancy way of saying that it refers to one species in the genus. It has a lowercase letter, and it's also written in italics. So why do we care about these scientific names? Well, you could come up with a lot of common names for an organism that vary based on location. Take this mountain lion, for example. It's also known as a puma, cougar, or Texas panther. Same animal, different names. But its scientific name here is specific and recognized regardless of your location. And that gives power to an awesome way to organize and name species. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, you guys, so that is the end of chapter 1. Let me know if y'all have questions.